Well, hello everyone, and welcome back to Human Biology here at Chaminade University. Today we are going to be starting Module 5B when we talk about the immune system. Now, what's the purpose of the immune system? So it serves several functions. It's going to be able to take extra interstitial fluid back from the interstitial space, and then it takes it through lymphatic vessels and returns it to the bloodstream. But before it does that, it's going to send it through filtration mechanisms in like the lymph nodes and lymph nodules that are going to destroy any of the foreign invaders that are found in that fluid. It's also going to serve to supplement the nutritional system by taking anything that is fat soluble, so any dietary fats or vitamins that are fat soluble, and moving them from the region where they're absorbed in the small intestines back into the bloodstream. So they can't go into the bloodstream directly, so they're going to have to end up going through the lymphatic system and then end up in the bloodstream after that. But the most important role, I think, of our immune system is to go ahead and attack anything that's trying to kill us. And as you may know, everything on the outside of our body is constantly trying to eat us. So the immune system is going to recognize infectious organisms like bacteria, fungi, viruses, parasites, etc., and slaughter them within our body. And in order to do this, it has to be able to recognize self from non-self. So self-tolerance is very important. So they have to be able to tolerate their own cells so that we don't attack our own selves, like in autoimmune diseases, for example. And they also have to be able to recognize what kind of foreign molecules are non-harmful, so just things like grass or other environmental substances that are present, right? And if we end up having this go aberrant, then we can end up with an allergic reaction to something that is a non-harmful foreign molecule. It also allows us what we call immunological memory. So once an immune cell sees something that it needs to attack, right, a foreign body, it then creates a reserve system of stem cells that can then create more of that cell in the future to be able to attack that similar organism if that organism invades our body again. Okay, so here's an overview of our immune system. As you are going to notice almost immediately, we have a very strong overlay with the lymphatic vessels, as with the cardiovascular system. So you can see it's going to service our limbs in similar regions to where we are going to have vascular service as well. It also is going to service here our gut. So we have a lot of these nodes. We have lymph nodes in our inguinal canals. We have lymph nodes inside our armpits as well as up in our throat. So one of the first things that we check when we feel if we're getting sick is going to be to put our fingers right here on the side of the throat and see if we have swollen lymph nodes, right? But we also have mucosal associated lymphatic tissue that's found all inside of our gut and our digestive system, which services twofold. One, to pick up those dietary fats and lipids, and secondarily, to be on site present if we do have some sort of foreign body that's going to infect or invade our system. All right, so what happens? So we have fluid that's being pushed out of the cardiovascular system to go into the interstitial fluid, and then that bathes the cells, and there's an exchange between the cardiovascular system, the interstitial fluid, and then the interstitial fluid with the cytoplasmic fluid, and then that exchange goes back from cytoplasmic fluid, interstitial fluid to the bloodstream. And there's some fluid that's going to be residual or left over. So that fluid, so that we don't end up with an edema, which is when we have swelling in certain locations, that fluid's going to get picked back up by the lymphatic fluid or by the lymphatic tissue and forced back through these lymphatic nodules, which are going to be sites where they're going to be T cells and B cells and macrophages and other types of immune cells that are present to digest any sort of parasites, viruses, microbes that may, or foreign invaders of any kind that may have come into our system. So after we do that, we force it through these little, I call them Genki sushi, where we're just going to force all this fluid through, and whatever's inside here is able to eat up all the foreign invaders it wants. Then that fluid's going to get dropped back into these ducts here, which are the lymphatic ducts. So we have the right lymphatic duct here, and we're going to also have the thoracic duct, which is the left lymphatic duct, essentially. Now, I want to take note of the fact that the drainage systems here are not exactly equal. In fact, they're incredibly unequal. This region here, almost 3/4 of your body, is drained by the thoracic duct in through the left into here, this region where we're going to have the jugular and the subclavian come together. That's where we're going to have that duct here. And here we have the lymphatic duct that's going to fit right here, again, also into the jugular vein and the subclavian vein. So yeah, this thoracic duct is going to feed right back into the body here. So again, 75% of the body is going to be drained by the thoracic duct or the left lymphatic duct, and the right lymphatic duct is going to drain that last 1/4. Okay, so again, that thoracic duct, which you can kind of see coming up here, is then going to drain into that subclavian vein just at the notch between the jugular and the subclavian. And it's the same thing here on the right side. All right, so if we go ahead and take a look at what happens during capillary exchange, as I mentioned, the lymphatic system is going to overlay almost directly with the cardiovascular system here. So here we have the arteries coming in. Remember, arteries to venules, arterioles to venules. We're going to have capillary exchange. So here's our capillary bed. This is where we're going to be losing oxygen. So you can see it's going from red to purple to blue. And the fluid, the reason it's losing oxygen is because it's losing a lot of fluid, and the fluid is just pouring out into this interstitial fluid, which bathes these cells and exchanges with the cytoplasmic fluid and comes back out. And again, most of that is picked up by the cardiovascular system and returned into the venules and returned back into the heart directly. However, a good percentage of it, or a small percentage of it, a small percentage of it is going to be picked up by these lymphatic vessels. So any overflow that would normally cause edema, we want to be able to have return. And the lymphatic vessels have these one-way slits. So we have openings where fluid comes in, and they also are going to have one-way valves, so we're pushing fluid out. And eventually we're going to go through the lymph nodes and then back into the cardiovascular system. Okay, so again, to avoid edema, which is when fluid can accumulate in the interstitial fluid, we want to utilize the lymphatic system to drain any of the fluid that's been pumped out by the cardiovascular system and not returned into venous return. Now, if we do end up with edema, there's many causes of this fluid buildup. It basically is going to happen because more fluid is coming out of the vascular system than is coming back in. And this can happen because we have an increased hydrostatic pressure in the capillaries, or we have abnormal kidney function, or failure of the valves in the veins. So we have one-way valves in the veins that keep things going forward. And if they don't continue to push everything forward and allow fluid to slip backwards, then we can have fluid that leaks back out, or fluid that's in the venous return system that therefore prevents new fluid from coming in. All of this can then cause edema. I may have mentioned when we talked about the cardiovascular system that a good bit of the fluid return in venous return, it comes from the skeletal muscle contraction system. So when you walk around in your lower limbs, every time you contract your muscles, you're squeezing the blood flow up past a series of one-way valves that then doesn't allow for it to return backwards. So it's eventually going to push its way up into the vena cava and back into the heart. But if you're bedridden and you're not moving around quite a lot, then that blood tends to pool instead of being pushed up through those one-way valves. And again, once the blood pools in the venous system, then nothing else can come in, or a limited amount of fluid can come back into the venous system, so edema starts to occur. This can also happen because of medications or lymphatic vessel issues, or a low amount of plasma proteins, etc. Anyway, what happens is we end up with edema, and that edema ends up causing swelling or accumulation of interstitial fluid, essentially. All right, so let's look at the different types of cells that we have in the lymphatic system. In order to do this, I'm going to take a moment to discuss some stem cells. So stem cells are very specialized cells. These are cells that, they're specialty cells, I guess, that have the ability to become many different types of cells. Stem cells are classified as either totipotent or pluripotent. Totipotent means that they can become any cell in the body. Think like embryonic stem cells, which can turn into eye cells or heart cells or liver cells or skeletal cells, right? But a lot of the stem cells in the adult body are what we call pluripotent. That means that they have a lot of potential, multiple potentials, but not every potential. Basically what that means is that if we are a lymphatic stem cell, we can become cells of the lymphatic cell line, but we're never going to become, say, a heart cell or a retinal cell. So that's pretty common with adult stem cells. So here's a pluripotent, so multiple potential stem cell called a hemocytoblast, and it can differentiate into multiple different lineages. Before we talk about differentiation, one of the other hallmarks of stem cells is that they always create a renewal. So they have self-renewal. So they're going to create a new stem cell as well as one that can go on to differentiate. So this cell will reproduce. It will have one cell that replenishes the stem cell lineage and another cell from that division that will enter into one of these pathways. So the first split is going to be into the lymphoid cell line, which is then going to become T lymphocytes and B lymphocytes, plasma cells, natural killer cells. And then the other cell line is going to be the myeloid stem cell line, which eventually will become myoblast cells, which can differentiate into different types of white blood cells, basophils, neutrophils, eosinophils, etc., or red blood cells, which are erythrocytes. And then we can also have mast cells, which are involved in the immune response. We'll talk about how in a minute. And megakaryocytes. Now, these guys are really interesting. So megakaryocytes are basically going to bleb off from each other and make these little teeny guys here, which become thrombocytes. They're going to be the platelets, basically, that are going to be involved in the clotting response. These are not cells. These are cell-like particles, just to be clear. But the megakaryocyte is a cell. So the megakaryocyte is a cell that gives rise to platelets. Okay. Now, where do these cells come from? Well, the short answer is bone marrow and the thymus. So when we're talking about B cells, they're coming from the bone marrow. When we're talking about T cells, they come from the thymus. They're going to start off immature, both B cells and T cells start off immature. The immature T cells are called thymocytes. And eventually they have to undergo a maturation process. Now, they both undergo maturation processes a little bit different, but the point of the maturation process is for them to undergo tolerance so that they become tolerant of self and intolerant of non-self. Therefore, they will react to foreign invaders, but they will not react to self. So they won't attack molecules that are of the person's body, right? All right. So I mentioned lymph nodes previously. Lymph nodes are regions where the lymphatic fluid is going to flow through, and then be eaten. Basically, all of the bad bacteria and viruses, etc., are going to be eaten by T cells, macrophages, B cells, etc., that are all hanging out. I like to call it little Genki sushi, where the fluid flows through, and everybody who's sitting there, like the T cells and B cells, can eat whatever they want from it. And so that's going to help clean out the fluid, so it's going to help fight infections by removing the foreign invaders. It's also going to help eliminate some toxins and also any dead cells that were carried in the lymph nodes. And this is why our lymph nodes tend to swell when we get a little bit sick, because we have a ton of dead cells that are going to end up eating that lymph, which ends up causing the lymph to swell, as well as the fact that we're going to have, during an infection response, an increase in the foreign invaders as well. So lymph nodes are found all throughout the body, basically in coordination with where you find your cardiovascular system, but they're found in much larger numbers in areas where microorganisms are likely to enter the body. So they're found in areas like the ears, the mouth, the lungs, your genital tract, digestive tracts, mammary glands, areas that are going to be accessible to the external environment. All right, this is what a lymph node looks like. This is what a series of lymph nodes look like, right? They're all in conjugation. This is why we tend to see swelling in the throat, because there's a lot of them present in that area, but we also have them in the face, in the back of the head, anywhere that you're going to see the cardiovascular system. We're going to be responsible for draining the fluid back out from anywhere that the fluid has been deposited. All right, so here we have both arterial flow and venous return into this lymph node. Okay, so we have blood supply, as well as having fluid that's coming in. So this is the lymphatic fluid coming in through what we call afferent lymphatic vessels. So afferent feeding in, efferent feeding out. All right, so lymphatic fluid coming into is going to then go through these regions where it's going to be digested by different types of cells, and then exit out as clean fluid through the efferent lymphatic vessels. Okay, so lymph nodes serve to filter out the lymphatic fluid before it is returned to the blood supply. Okay, so other regions of the lymphatic system are going to include your spleen and lymphatic nodules. So let's talk about the lymphatic nodules first, because they're very similar to lymph nodes. So we talked about lymph nodes just a second ago, except lymph nodes have a capsule around them. Lymphatic nodules do not have a capsule around them, and they're found in regions that are, again, going to be exposed to the external environment, like your tonsils and your gastrointestinal tract, right, and your intestines. These are all going to be areas where you're exposed to external environment, meaning that we're going to possibly be exposed to external microbes that have come in, and so we're going to be digesting anything that's present. Generally, it's going to be it's called mucosal associated lymphatic tissue, or in this case, gut associated lymphatic tissue, so MALT or GALT, all involved in the digestive system, as well as these specialty Peyer's patches found in, again, the digestive system and the small intestines. And then we have regions of the tonsils and the adenoids, which are other lymphatic nodules within the body. All right, so let's talk about the spleen. The spleen has two different types of pulp. It's got red pulp and white pulp. All right, so red pulp is going to have red blood cells, that's why it's so named as red, as well as platelets and macrophages. This is going to be an area where we're recycling our red blood cells. So the old red blood cells are going to come to this graveyard to die, basically, but really they're digested and recycled into new red blood cells. We also have white pulp, which is an area that's going to have a lot of white blood cells like lymphocytes, remember T lymphocytes and B lymphocytes, macrophages, dendritic cells, etc. And all of these are going to be involved in what we call the adaptive immune response. Now, we have a native immune response and an adaptive immune response. The native immune response is something that you are innately given an innate immune system to, so you're able to have a broad attack on foreign invaders. But a specific immune response requires the adaptive immune response, something that you've seen before, that you have created a specific arsenal to attack this particular foreign invader, and then when that foreign invader comes again, you already have the arsenal available. That's called the adaptive immune response. And this is what prevents you from getting sick from the same cold twice, for example. So this here is the spleen. As we see with almost every organ, we're going to have vasculature. Actually, with every organ, we're going to have vasculature. So here's a pretty large artery running in, supplying oxygen, and then the venous return. So there's a splenic vein. On the outside, we're going to have a capsule that surrounds the whole thing. And then we have two different regions, the red pulp region and the white pulp region. Now, the marginal zone is going to be the region between the white pulp and the red pulp. Here we have, this is called the periarterial lymphoid sheath, which is going to be white pulp as well. Here we have primary follicle, also white pulp. And then this here, again, that's the red pulp. So this is where the red blood cells are going to be recycled and created anew, essentially. And then again, the white is going to be where we have different types of lymphocytes and other white blood cells. Okay. So if we consider that Harold has been exposed to the common cold virus and is starting to show symptoms. He is exhibiting a runny nose, a sore throat, and has been coughing. So obviously the virus has gotten past his first line of defense, and so now we have to activate the acquired immune response. What are the series of events that have to take place in the immune system after the virus particles have entered his upper respiratory cells? And once his immune system attacks and eliminates the infection, can Harold become sick with the same virus again? Why or why not? And we just discussed that adaptive immunity, but we'll talk about that at the end. And what are some things that a patient might be able to do to increase their ability to fight off future colds? All right, so again, we have two major types of immune responses. We have the innate immune response, and we have the adaptive immune response. The innate immune response is going to be the first line of defense against foreign invaders, and really that's going to be skin and your mucosal membranes. So your skin is going to be the first protective mechanism to foreign invaders, a physical barrier that is going to be waterproof, and we're able to wash it off. So we are hopefully able to wash our hands before we touch our face or any other mucus barriers. And we also, again, have mucous membranes anywhere that we are facing the external environment. That includes the digestive system, the reproductive tracts, the respiratory system, etc. And in these mucous membranes, we're going to have specialty types of cells that are meant to attack any foreign invaders. So first of all, the mucus is nice and thick, which means that it'll collect and capture anything that ends up trying to come in with any luck. That means we might be able to expectorate it out. This is why we end up with a lot of mucus when we get really sick. Things like a bunch of snot that we're going to have to blow out, or a bunch of phlegm in our throat that we have to expectorate. Why? Because we upregulate the amount of mucus that we are going to be creating when we feel like we are under attack. Again, we also have specialty immune cells. They're found not only throughout the layers of the skin, but also throughout the majority of the body. These guys are meant to detect foreign invasion and destroy invaders. Keep in mind there are specialty cells that are meant to attack bacteria, different cells meant to attack viruses, different cells meant to attack fungi or parasites. So just like we would have different types of police officers for, say, vice versus homicide, we would have different types of immune system cells that will attack different foreign invaders. But either way, once these foreign pathogens enter the body, the immune system is going to throw up some red flags stating that it's detected foreign proteins. And when we're able to detect the foreign proteins, again, these are called antigens. So we have antibodies in our system that detect antigens. And when the antibodies and the antigens interact, this is going to throw up a red flag. Antigens don't necessarily have to be foreign proteins. They can be lipoproteins, glycoproteins, peptides, carbohydrates. They can be a lot of different types of biomolecules. Now, these are all proteins. Obviously, this one's a sugar. And all of these can be things that we recognize. And again, the main point here is to differentiate self versus non-self. Anything that is self, we want to allow to live. Anything that is non-self, we want to attack. Now, we also have chemical defenses, particularly on the surface of the skin. So things like lysosomes that are present in tears and in sweat. So these are going to attack the walls of many bacteria. Now, not the bacteria that are already present inside our body. So normal bacteria gets a pass, but foreign bacteria we do try to attack. We also have glands that are going to make the surface of our skin very acidic. It's the same thing with the female reproductive tract. The vaginal secretions are very acidic. Why? These are openings to the external environment, and we want to make sure that we're killing anything that's going to try to invade our body, right? So again, because the linings of the digestive tract and the respiratory tract, as well as the reproductive tract, these all provide routes that microbes can take to get into our body. And so we have to have chemical weaponry that we can try to prevent the infection with. So again, we have the lysosome that's found in our saliva as well as our tears. Inside our digestive tract, particularly in our stomach, we're going to be secreting hydrochloric acid, really low pH, so hopefully kill off any foreign invaders. As well as that, we're going to also have digestive enzymes that are going to hopefully protect us from any foodborne illnesses. Inside our throat and in our nasal passages, we have sticky mucus traps. Again, these are going to help take most of our microorganisms that enter into the body and attack them, or at least capture them so they can be attacked. And then we're going to expectorate this mucus. So we're actually going to cough this mucus up out of our body using cilia, continuously moving that mucus forward and out of our body. Same thing with the nasal membranes. Again, we're going to be getting rid of any of the mucus that seems to have caught a bunch of foreign invaders. Now, keep in mind that not all bacteria are bad. We also have friendly bacteria or resident bacteria. These are flora that are normally found in the digestive tract. These are really important for several things, including creating some of our vitamins, as well as digesting our insoluble fibers, etc. So these guys are going to play a very normal role in the digestive tract. And also their presence is going to help keep away foreign invaders, because picture that they've basically already taken up all of the housing space, so there's nowhere for these abnormal bacteria to move into. So the resident bacteria, basically by keeping their foot down and keeping a foothold inside your intestines, they keep away some of the foreign bacteria and help prevent foodborne illnesses and infections. Okay, so again, the skin and the mucous membranes are our first line of defense. Our secondary line of defense is going to be those chemical molecules that we talked about previously, cellular and chemical defenses. So anytime we have an inflammatory response, we're going to increase our temperature as well as increase blood flow to the area, which is going to bring in cells that kill the invading microbes. Some of them are already present in the skin and just kind of send up a red flag to bring in more. Some of them are going to come in through the blood supply. And we also have proteins that are going to kill invading microbes. So we have a lot of cellular defenses and chemical defenses that are going to play a role in protecting our body from foreign digestion. Okay, so these are different types of immune cells, and we're going to talk about what their role is within the immune system. So a neutrophil is going to be the most common white blood cells, and these guys are going to play a role in the inflammatory response. So they're going to produce histamine. So anybody that's ever had an allergic reaction knows that you take Claritin, right? That's an antihistamine. So if this whole process goes awry, again, mast cells are also going to release histamine. So when we end up with an infection, we end up releasing histamine. And sometimes we end up with this response going awry because we've just detected, grass, for example, or pollen, hay fever, right? And so you'll take that Claritin, which is an antihistamine, to slow the roll of this response. But under the normal physiological responses, these guys are going to be involved in releasing histamine, vasodilators, etc. So these chemicals are going to cause inflammation, and causing inflammation is going to bring more blood supply to the area, increase the temperature, which should hopefully kill off some bacteria, as well as increasing that blood supply is bringing in a bunch of, police officers or cells that are going to attack the foreign invaders. Neutrophils are also phagocytic cells. What does that mean? That means that they wrap their little arms around a microbe and engulf them and actually ingest and then destroy different types of microbes, particularly bacteria and other types of pathogens. And different pathogens that have already been labeled by the B cells for destruction, essentially. These guys come out of the blood vessels towards an infected area. Again, once we have the infection response, we're going to end up with an increased blood supply, as well as different red flags that's going to cause these neutrophils to come over. They're attracted specifically by damaged cells, by what's called chemotaxis, which is like coming towards a chemical signal. So their immune cells are going to migrate towards the area of infection because the infection is releasing a chemical signal that's going to call those neutrophils over. Okay, so we just went from the most common to the least common. The least common is called a basophil. Again, it's going to be responsible for releasing histamine involved in the inflammatory response, as well as cytokines involved in the inflammatory response. But as I mentioned, every police officer kind of has their own subspecialty. Well, this basophil is going to be involved in parasitic infections, particularly things like ticks or lice, or what we call ectoparasites, parasites on the outside of our body. Eosinophils are going to be involved in internal parasitic infections like worms and helminths. It's going to be responsible for creating chemicals like histamine that are involved in the allergic response. You'll see that this is a common thing. The allergic response is going to increase inflammation, and increasing inflammation increases temperature and blood supply, which allows us to attack the foreign bodies in that area. So this is why an area that is infected is going to get very red and swollen, because we're increasing the blood supply as well as the temperature. All right, so mast cells are also involved in allergic reactions and what we call anaphylaxis, which is when you have a difficulty breathing. What happens here is we're going to have an increase in the histamine response when we are presented with specific antibodies. So the antibodies against XYZ, we're going to then cause histamine release, which can sometimes cause anaphylactic shock. So allergic reactions have a risk of anaphylactic shock, which is when the individual is unable to breathe. It's going to close off the airway, etc. This is why we're going to need an EpiPen for someone who has, say, a peanut allergy or some of these more severe extreme allergies. Nut allergies are pretty extreme. Fish allergies are pretty extreme too. So anaphylactic shock is basically when this system goes awry, right? When we get exposed to something that really shouldn't be harmful to us, but we go overboard attacking this and actually cause our own system to kind of go into shock. And at that point, we're going to basically be going into overdrive of this allergic reaction. But under normal conditions, it's going to play a role in releasing histamine to cause the immune response, and then also play a role in wound healing and protective healing. And it also is going to be found mainly in areas of the body that are open to the outside, like your nose, your skin, your gastrointestinal tract, your lungs, etc. All right, monocytes are also responsive to inflammatory signals. And these guys are going to come out of the blood supply, so extravate, migrate into the tissue, and then eventually differentiate into macrophages or dendritic cells. So monocytes are going to be able to differentiate into the macrophages that we haven't talked about them yet, into dendritic cells. Oh, here they are, dendritic cells and macrophages. So what's the difference? Okay, so these guys are both going to be antigen-presenting cells. What does that mean? That means that these guys are going to take an antigen from a foreign invader and present it specifically to B cells and T cells to stimulate their response. So it says, hey, I found a foreign invader. Here's a piece of it. Looks like this. It's the equivalent of giving your bloodhound the scent so that then they can go and find the foreign invader. So they're going to stimulate these bloodhounds to go be able to attack whatever the foreign invader is. Dendritic cells themselves are also phagocytic. That means they're able to engulf pathogens and pull them into and digest them inside their cells. And again, typically they're present in any tissues that are in direct contact with the environment, your skin, your lungs, gastrointestinal tract, etc. Macrophages, also antigen-presenting cells, and these guys are going to stimulate lymphocytes specifically. So dendritic cells stimulate T cells and B cells, right? Lymphocytes. Macrophages also are going to stimulate these T cells and B cells and initiate what's called the adaptive immune response. So giving them a target to attack by giving them the scent or the antigen to know what they're going to go after. Macrophages, again, they're also phagocytic cells, can engulf pathogens, etc. But the major difference here is that they're also going to be secreting toxic chemicals that can directly kill the invading organisms. So they don't have to phagocytose them, they can actually secrete toxic chemicals. And on top of that, not only are they secreting chemicals that are toxic to the foreign invader, they're secreting chemicals that are directly involved in the inflammatory or autoimmune response, right? Cytokines and other chemicals that are basically going to send up red flags for other immune cells to be able to extravate out of the blood supply and come and find the area of infection. Okay, so last but not least, let's talk about natural killer cells. As I mentioned, the entire role of the immune system is to differentiate between self and non-self. So the role of the immune system is to kill non-self and to let self survive, with the exception of the natural killer cells. These guys are the police of the police, if you will. These guys kill cells that are self. Cells inside of the body that have been infected by viruses, for example, or cancer cells that are going to be undergoing cellular division that's completely unchecked. And so what do? They specifically attack self, so cells that are your human body self that have been infected or become cancerous, by using special T molecules called porphyrin, which creates holes inside the cell, or they also can induce what's called apoptosis or programmed cell death, Remember, this differs from necrosis because necrosis is just going to end up with everything dying without any use of any of it. Apoptosis is going to be a disassembly of the cell so that all of the parts of that cell can be utilized by the surrounding cells prior to the cell death. Natural killer cells also are going to kill any cells that have already been what we call opensized with antibodies, so they've been labeled for cell death essentially. And they also have the ability to have the acquired immune system, so they can produce memory cells that can detect the same infection if it comes around again. So these guys can have a specialty subset of cells that lingers for many years to be able to wait to see if that foreign invader attacks our body again. All right. So again, two major types, acquired and innate immune responses. So first we're going to start with the innate immune response. This is specifically talking about natural killer cells finding a target cell that is going to be targeted for apoptosis for whatever reason. So once the target cell has been bound to, and this target is going to have labels on it somehow, this killer cell notices that it's not a normal self. It is still self, but it is an abnormal self cell. So once we've attached ourselves to the target cell, we're going to have these granules lyse open, releasing what's called perforin. Now, when the perforin is going to hit the target cell's plasma membrane, they cause this weird granularization. So the perforin is going to cause a shape change, and a small pocket of intracellular space in between the plasma membrane is going to open up. Then we're going to have calcium exposed to that area, which causes the remodeling of the perforin molecules. Long story short, we end up with holes in the membrane that allow liquid to flow right on through these pores. The pores are going to emit salt and water with no discrimination at all, causing the target cell to swell up. Water is just going to flow basically in until it eventually bursts open. So that means that this cell, which was a cancerous cell or an infected cell, etc., is now going to be targeted for cell death by these natural killer cells using these perforin molecules. All right, so cells that are infected, they can be infected by a virus or a bacteria or a microbe or whatever, they typically are going to make a chemical called interferon. And interferons are found inside the cell as well as secreted into the surrounding area. And you can kind of picture this like a neighborhood watch where one person in the neighborhood got robbed, and then they tell everyone else in the neighborhood, hey, I got robbed last night. Maybe you guys should pay attention. What do I mean by that? This cell is infected. Hey guys, I'm infected by a virus. You guys should pay attention because there might be viruses that are around here. In fact, there clearly are. I got infected. And on top of that, just to kind of let you guys know, heads up, I myself am going to be creating a bunch of viruses, lysing open and secreting those viral particles. So it's kind of like an SOS that gets sent out, a distress call, if you will, but more of a warning signal that gets sent out before the cell is going to lyse open and release a other bunch of viruses to the other cells in the area. So once interferons are formed, the cell is going to secrete them to the surrounding cells to be warned of the potential of viral infection, and so they're going to produce proteins that are able to inhibit viral replication if they are infected, right? The equivalent of arming yourself knowing that you might be robbed, right? So these interferons are going to activate natural killer cells in the infected area. So again, we're going to be mounting an immune response as well, and they're going to activate something called P53. Now, I don't know if we've talked about P53, but P53 is a very powerful oncogene, which is going to be responsible for increasing the cell the cell replication. So it causes a signal that causes the cells to continue to replicate more quickly than they would normally without going through all their checkpoints. Also, these interferons are kind of what you feel unwell. When you start to feel kind of sick, when you're like, I feel like I'm infected, I might be a little bit groggy, I'm not feeling well. These interferons are created by bacterial toxins, and they don't feel good. So they kind of let your body know, hey, you should rest. Okay, so here's kind of what happens. The virus infects a cell. This cell here is the equivalent of this home getting robbed, right? Once this cell gets infected, it's going to create interferons. The interferons are going to be released, so created inside the cell and released by the cell. Now remember, this cell is under attack. It's actually actively creating viruses and will eventually lyse open and release those viruses. So it's doing its neighbors a favor by saying, hey, I'm infected. You should upregulate your defense mechanisms. So once we end up with the interferon binding, Then we're going to activate the virus blocking enzyme so that when the virus does, or if the virus does attack and try to invade, the interferon response has already caused virus blocking enzymes to prevent viral entry. So therefore, even if the virus gets in, it's not going to be able to replicate because the virus blocking enzymes have been activated. Okay, so interferon, again, is going to be basically like the neighborhood watch where it tells all of the cells around the infected cell, hey, watch out. It's too late for me, but run and save yourself, right? Okay, so a lot of this works by what's called the complement system. These complement systems are going to be proteins. There's approximately 20 of them. They circulate throughout the plasma. They're created by the liver, circulate through the blood plasma, and they start off inactivated. So basically they stay unactivated until they encounter a foreign molecule, like a fungal cell or a bacterial cell wall or something like that. As soon as they do, they form a membrane attack complex. So a bunch of these different complement proteins come together to create an attack mechanism that forms a hole or a pore in the foreign cell's membrane. And just like we saw with perforin, when we get a hole in the membrane, water rushes in, the cell bursts open, right? And so this complement system is going to serve as a way that we can target and attack cells that are foreign invaders, like fungal or bacterial cells. And we also do this by triggering by antibody response. So we use antibodies to recognize antigens that are found on the fungal or bacterial cell surface, and then activate that complement system to be able to know, oh, we have fungal or bacterial cells here, so now we should go and find them and attack them. It also is going to help facilitate phagocytosis. Again, phagocytosis is when we're going to engulf a foreign body or a foreign cell, bring it into, so we invaginate it into the cell, and then digest it once we bring it in. Okay, so again, here's an example of what occurs during the immune response. So here we have, I don't know, what is this, a splinter of some sort of break in the cell, in the skin that's going to actually go all the way through the skin layer into this, called an injury site, okay? That, unless this cut has happened from a scalpel in the OR, it's very unlikely that this is perfectly clean. So more than likely this, whatever it is, looks like a splinter, will introduce a bunch of microbes or bacteria at the site of injury. Okay, so when the bacteria get in there, and they first get in there, remember bacteria replicate in the order of every 2 hours, and this is a nice warm environment filled with sugar. The bacteria are in heaven. This is exactly where they want to live, right? They want to eat you alive. So what do we do to prevent them from doing this? Well, we have a lot of different types of cells that are going to work together. So when the break in the skin introduces bacteria, they start replicating like crazy at the wound site. That activates macrophages. Macrophages recognize that the bacteria are present. They start engulfing them if they can, right? So they're going to start phagocytosing as much as possible, but we just can't eat them as quickly as they reproduce. So we also are going to call for help by creating cytokines, right? So these activated macrophages engulf the pathogens as much as they can and create cytokines and chemokines, which are going to activate the inflammatory response. We also have these mast cells here, and mast cells are going to recognize that there are bacteria present and throw up a red flag. That red flag is in the form of what we call histamine. Again, this is the allergic response. In this case, a perfectly normal allergic response because we want to get rid of this bacteria. Now, if we were throwing up histamine because we had pollen or something, then we want to take some Claritin to tell that histamine to chill, to slow down. But for now, we want that histamine present because the histamine is basically saying we have an infection present. So what does it do? Histamine and then all those cytokines released from the mast cells and the macrophages are going to make it so that the local blood vessels are going to dilate. That means that we're going to have spaces in between where we can have extravasation of neutrophils. We also are going to have specialty cytokines right here that are going to go along this region, making the blood vessel wall sticky. That means that the neutrophils are going to bind to those cytokines, again, indicating that this is an area of inflammation or under attack. The neutrophils are then going to extravate out through those little holes. So the neutrophils pass through the cells and the blood vessel wall and migrate into the infection site where they also begin the process of phagocytosis. So all of these cells work together. The macrophages are an immediate immune response. The mast cells throw up the red flag known as histamine. Macrophages get to work ingesting, but they also need to send out a chemical signal, which is going to call neutrophils to the scene, and the neutrophils then do their thing and ingest as much of these as possible. Okay? So again, mast cells and basophil cells release histamine. They also are going to release prostaglandin when tissue has been damaged. That causes vasodilation, which causes the capillaries to become more leaky or more permeable, which allows blood cells, white blood cells particularly, like the neutrophils we were just talking about, but also mast cells and macrophages, right? All these white blood cells are going to be able to enter into the tissue. They are going to, in concert, cause redness and swelling. Why? We're bringing in more blood supply, and we are bringing in more fluid, and also we're also going to be upregulating our temperature. So we're going to increase our temperature here too. So we're going to see a fever basically, or at least a localized temperature response. Now, prostaglandins are going to cause pain sensation, which is going to cause us the aching feeling that we get when we're sick. So prostaglandins also make us to feel unwell. And as we mentioned, the neutrophils and macrophages have a role in killing the microbes by engulfing the dead cells. Now, when we have a site of infection, we can see pus, right? What is pus? If you look at pus under the microscope, it's going to be a bunch of dead cells. Dead cells that have been attacked and have lost the battle against the foreign invaders, as well as white blood cells that have been ingesting these foreign invaders as much as possible, but again, lost the battle. So these are our dead soldiers, essentially. And so this is why the symptoms of inflammation include redness. Well, we brought more blood supply to the area. Pain, well, that's the prostaglandins activating our pain receptors. Swelling, again, more fluid to the area. Heat, well, that's part of our response as well, right? So we're going to be our inflammatory response as well because we want to kill off foreign invaders. And that's also going to, all of this causes a reduced function of the area. Okay, so when the macrophages are going to attack, they're going to cause an increase in your body temperature. Why do we do that? Why do we cause a fever? Because the bacteria are very temperature sensitive. And if we go too hot, we can kill the bacteria. So a fever can curve, and maybe if it doesn't kill them, it at least slows their growth. And if we can slow their growth enough that our immune system is able to actually kill off the bacteria, then that's a win-win. So fevers are going to be beneficial to a certain degree, but can be extremely dangerous if the temperature gets too high, because that's going to start inactivating cellular enzymes. Remember, enzymes also have a certain range in which they're going to be working, and critical enzymes are responsible for, well, everything, including brain function. So if we get a fever that's too high, right, over 42 degrees Celsius, that can be fatal. Another reason that we end up with this fever response is because certain pathogens are going to act as what's called pyrogens. So pyro meaning fire, genic meaning creating, so creating an increase in temperature. Again, all of this leads to what we call the fever response. Now, a fever up to a certain degree, very helpful. Anything over that, we should be treating, and then this will be like Tylenol, etc. And then a fever over certain degrees can actually cause brain death. Okay, these are different cells that are involved in the immune response. So these are B cells versus helper T cells, cytotoxic T cells, and regulatory T cells. B cells come from the bone marrow. T cells come from the thymus. All right. B cells are antigen-presenting cells. That means that they're going to take some sort of piece of a foreign invader, an antigen, just a part of it, present it to another cell that's giving the bloodhound something to sniff so that it's able to know what it's targeting, and then send it off to kill whatever it is. So B cells are able to be phagocytic when they are actually finding their target cell. They can engulf it and kill it themselves, as well as be an antigen-presenting cell, which can cause the immune response. They also are going to produce cytokines. Again, we just talked about those right here as something that's going to be released here by the macrophages, again, causing the immune response. And the B cells differentiate into plasma cells when they're activated by what's called the helper T cells. We just talked about those. And then once they activate into plasma cells, then they secrete antibodies. And antibodies are part of the immune response because antibodies go out and find antigens and target things that have that antigen for destruction. Also, keep in mind that these cells are able to have memory cells. Memory cells are a subset of the cells that are kept for the next time that foreign invader tries to invade your body. This is a way that we're trying to prevent an individual from becoming sick from the same disease again. So if we know that last time this arsenal worked against this foreign invader, we keep that arsenal at the ready, essentially, so that the next time that we see that foreign invader, we're able to upregulate and kill it so we don't end up getting sick again. Okay, helper T cell, well, T cells are also able to produce memory cells. So all of these cells are able to create a subset of cells that they can store for the next immune attack. All right, so T cells, we have several different types of. We have helper T cells. These guys are not directly involved in killing the invading organisms, but they're antigen responding cells. So when an antigen is presented, like by the B cells or a macrophage or a dendritic cell, they produce a response. And when they produce the response, they produce cytokines. Cytokines are going to activate these B cells. So again, they go kind of hand in hand. Also going to activate the cytotoxic T cells, as well as macrophages, dendritic cells, and natural killer cells. So these helper cells, these helper T cells, are going to recognize that there needs to be an immune response, because they have some sort of antigen that's been presented to them by antigen-presenting cells, and then they're going to create a bunch of chemicals that are going to activate B cells and cytotoxic T cells, et cetera, to upregulate the immune response. Now, cytotoxic T cells actually go out and do the dirty work themselves. They kill virus-infected cells, cancer cells, transplant tissue, so they kill self that is abnormal. Right? Again, these are considered to be self. This is going to be virus-infected cells, so it's cells that are self but are infected. Cancer cells are self cells that are reproducing out of control, and these are non-self that we're expecting to be treated as self, and so we'll have to take immunorejection drugs pretty much forever after an organ transplant to prevent the cytotoxic T cells from attacking the transplanted tissue. So when we have a transplant failure, it often has to do with this immune response or an autoimmune response. All right, so regulatory T cells are going to inhibit immune cells, other immune cells, as the infection starts to wind down, to help kind of stave off the infection and end the infection and prevent autoreactive immune cells from damaging tissues. Okay, so that's all the innate immune response. The adaptive immune response is a little bit different. The adaptive immune response is going to be cells that are going to acquire learning and memory, if that makes sense. Cells that have been exposed to a foreign invader, and while they might not have known immediately what to do with it, they found a weapon that worked, something they could put into their arsenal, and then they stored it for later. These are going to include B cells, plasma cells, and different types of T cells, including helper, cytotoxic, and regulatory T cells. So again, all the same cells that were involved in the innate response are also going to be involved in the adaptive response. The adaptive immune response depends entirely on antigen recognition. So we basically are going to have pathogens that are either present or non-present, and monocytes that are constantly looking for pathogens. When monocytes encounter that pathogen, then they turn into macrophages. So monocytes are a precursor to macrophages. This is like a soldier that hasn't yet gone to war. After they go to war, they change a little bit, right? These macrophages have a specific job now. They know how to respond to this foreign invader. Now, macrophages are going to live for an extremely long time, months, sometimes years, whereas neutrophils, which we just talked about, are only going to live for a short while, just a couple of hours. So macrophages, because they have a longer lifespan, have a little bit more of a memory, for example. So macrophages are going to recognize pathogens by pathogen-associated molecular patterns. What does that mean? Regions of the cell that don't look like human cells, okay? Pathogen-associated molecular patterns. So when we see a PAMP that does not look like a human PAMP, it looks, well, that would be called a HAMP, right? So it's a pathogen-associated pattern. Then we can identify it, and after that, we can phagocytose and digest the microbes. And after we phagocytose and digest them, then we regurgitate pieces of the microbe proteins to alert other immune cells. Again, this is called antigen presentation. So we previously talked about antigen presenting cells, and now we're going to talk about how antigen presentation works. So essentially, we're going to have antigen presenting immune cells, like I said, macrophages, etc., which once they encounter a foreign body, they digest it by phagocytosis and then take little pieces of it and put it back on the surface to present the smell of it, essentially, the antigens, to the B cells and the T cells, right? The bloodhounds that are going to go out and attack more of those foreign invaders. It says, all right, I killed this one, but you guys need to go out and get the rest of them. This is what it smells like. This is what it looks like, by utilizing the antigens that they've presented from the one that they've digested. Okay, so here we have a microbe here. It's got different antigens on the surface. It's going to be engulfed and then digested by this macrophage, right? So once this macrophage digests it, then it's going to take pieces of it and pop them into these MHC2 molecules, which are going to then be excreted or protruded from the membrane, so that T cells and B cells can come have a sniff at this antigen and see what this antigen looks like, and then go attack anything that has the same scent, right? So again, this is the equivalent of giving the bloodhound the dirty sock of the person they're trying to find or whatever. It gives them an idea of what it is that they're going to be targeting. Okay, so let's talk about T cells and B cells. Again, these are lymphocytes. They are white blood cells. These guys are going to be part of the immune response, and they originate in two different parts of the body. T cells originate in the bone marrow, and B cells, T cells are going to start in the bone marrow, but eventually migrate to the thymus. So their origin, let me back up. Their origin is the bone marrow, but eventually they go to the thymus gland, which is where they mature during embryonic development. So they're called T cells because they come from the thymus gland. B cells, on the other hand, are going to stay in the bone marrow, complete their maturation in the bone marrow. And both of these guys are going to be cells that are going to recognize antigen-presenting cells, and then take that antigen and remember that antigen and then go attack that specific antigen. So they recognize microorganisms by the antigens that they've been presented by antigen-presenting cells. So these guys are the bloodhounds, right? And then they go out and attack them. These are different in a couple of ways. One, the B cells are going to mark the cell for destruction, but not kill the cell itself. So it's basically going to be coating the cell or the antigen that's on the outside of the cell and marking that cell for destruction. Whereas T cells are actually going to go out and do the dirty work themselves, recognize and slaughter different microorganisms, viruses, by figuring out which antigens they need to attack based on, again, what the antigen-presenting cells have presented. Okay, so the T cell response is called the cell-mediated response. Basically what happens is we have to have a signal that's sent up, and again, that's in the form of like cytokines, etc. So when T cells secrete the bat signal, right, cytokines, that activates B cells, cytotoxic T cells, natural killer cells, macrophages. It activates all of the different immune cells, which again, these are all like different police officers that will attack different foreign bodies. And we have two different types of T cells here that we're talking about. CD4 positive cells are the helper T cells, and CD8 positive cells are cytotoxic T cells. You don't need to remember that they're 4 positive or 8 positive, but you do need to know the difference. So T cells, helper T cells, are going to secrete cytokines that are going to target the cells for destruction by activation of these other types of cells. Cytotoxic T cells actually go out and destroy the virus-infected cells. So these are the ones that are actually going to go out, boots on the ground, find the foreign body, or in this case it's a self that has been attacked by a foreign body, as well as cancer cells, again, abnormal self, and transplanted tissue, which we're trying to avoid by having that anti-rejection drugs. So it can also attack non-self, that is mimicking self, and it is going to destroy it using perforin. Remember, we talked about perforin and how it's going to make pores inside the cells. Now, regulatory T cells or suppressor T cells are going to be responsible for inhibiting the B cell and T cell response. Basically, this is going to say, hey, the fight's over, guys, go home. And then we also have cytotoxic T cells. Remember, we talked about the CD8 positive cytotoxic T cells. These guys are going to fight intracellular infection just by killing the cells that are infected. So instead of killing like the microbes that are infecting the cells or the viruses that are infecting the cells, they're just going to kill the cells that are actually already affected again. So we're going to find self that is not behaving properly. Okay, so here we have the MHC, or master histocompatibility complex, 1 and 2 expression, and which cells are going to express them. So MHC1 are expressed in all body cells with the exception of red blood cells. And the ones that react to it are going to be cytotoxic T cells. Again, they only react to cells that are infected with a virus or cancerous cells or something that's abnormal about them, right, transplanted tissue. And then we have MHC class 2, which are the types that are going to present antigens. So these cells will first kill one, basically take a sample of the foreign invader, kill one, digest it, and then break it down into its antigens, present those antigens on the surface of the cell so that other cells like helper T cells, etc., are able to react to it and then go out and find the foreign body that is going to be similar to this. Okay, so again, APC stands for antigen presenting cell. Here we're going to have what's called phagocytosis and antigen presenting. So this is the first time that we have encountered this pathogen. Okay, and so we have these particular regions on the pathogen, the PAMP, pathogen-specific biomolecules, that are going to be, so once this pathogen gets digested, so first we're going to phagocytose it, bring it in, then we digest it down into its components, and then some of those components are going to be presented by the macrohistocompatibility complex 2, on the surface of the antigen presenting cell. So this is basically saying, okay, I took a sample of this foreign body and this is what it looks like. And then we take it over to other cells like helper T cells and we tell them this is what it looks like, go get it. So this is the bloodhound. And then once the bloodhound has the scent, which again it's going to get by whatever's presented as an antigen, then we're going to create more and more and more of these particular cells that we can use to go out and fight the foreign invaders. So now these guys have a specific scent or target they're going to go after. These bloodhounds are going to go find more of this pathogen. At the same time, we're going to be creating memory cells, which are going to allow us to store this arsenal that we've just created for the next time that we encounter this foreign body that's attacked us, so that we can make sure that we're able or we're armed and ready for the next immune attack, right? All right. So if a macrophage is going to, well, once it phagocytoses and digests the foreign antigens, then it's going to present the foreign antigens on the surface of the cell to trigger the cell-mediated response, at which point T cells become activated and start secreting something called interleukin-2 or IL-2. And IL-2 is going to create more cytotoxic T cells. Cytotoxic T cells go out and destroy your infected body cells, again using perforin, which causes holes in the membrane of the cell and then causes cell swelling and lysis. Cytotoxic T cells are responsible for killing cells that are infected, so self that is not acting properly. So therefore, and basically they're going to run up a white flag up their flagpole that says, hey, I'm displaying this particular antigen having been affected, so I'm infected. And that means that cytotoxic T cells can target them and kill them, as well as we have special T cells that are going to be proliferative and continue to clone themselves. Right? These special cytotoxic T cells continue to clone themselves until the viral infection is completely taken care of. We also, again, are going to keep a memory of this arsenal, whatever weapon it is that we were able to use against this foreign invader, so that if we see the foreign invader again, we can attack them right away without letting them proliferate too much. Okay, so this is what the cell-mediated response looks like. Here we have a cell that's been virally infected. Okay, it is going to send out different fragments from the virus on the surface of the cell. So we have an infection, the cell knows it's targeted for death, but it wants to make sure that the cells around it know what it looks like. This is akin to taking the picture of the person that robbed you so you can show everyone in your neighborhood. So they basically are going to present the antigen on the surface of the cell so that the T cells are able to recognize it. Then the T cells are going to do a couple of things. One, they're going to proliferate. So we're going to make a lot of them. So we're going to have a bunch of clones. Again, one of them is going to become a memory cell that we can utilize later. The others are going to go out and become the foot soldiers, where they can go and actually attack any cells that are infected by this virus. Once we have this region, right, of this T cell receptor is going to bind specifically to the antigen on the surface of an infected cell, releasing the perforins and causing the cell to self-destruct, essentially. So this is a way that we can target self that is not acting normally, right? A normal self doesn't present these antigens because it's not under infection. So this is going to be able to target this cell for degradation while leaving the healthy cell right next to it alone. Okay. Now, let's talk about T cells a little bit more. So we have specialty helper T cells. Helper T cells are going to differentiate into special subtypes of helper T cells, including Th1 and Th2. Th1 cells are going to activate cytotoxic T cells. This happens typically in the case of a viral infection. Th2 is going to be responsive to a bacterial infection by activating B cells. As I mentioned, sometimes every time that we create a new weapon for our arsenal, we put it in our memory bank. So some of the helper T cells will be creating memory cells that will linger for a while to be able to help prevent a future infection of whatever it is that's infecting us now. And some of the helper T cells become regulatory T cells that go out and actually attack, so they're called Th17. They go out and attack this foreign body, so they're going to actually be responsible. So they're going to slow down the immune response and prevent any reactions against self. So they're going to be regulatory cells that are going to be responsible for slowing down the immune response. And some of them are going to become tolerant, or Th3 cells, to make sure that they're not reacting to normal things that we are exposed to that are harmless environmental molecules like food, normal floral bacteria, etc. Now, B cells can differentiate as well. So these guys are going to be differentiating. So humerals is going to be from the bone. Differentiate into plasma cells, and that happens when an infectious organism is going to cause this immune response. And once B cells are going to attack that infectious organism, they secrete antibodies. Now, again, this is a specific response, so an adaptive immune response. And every time that we have an adaptive immune response, we're going to take that arsenal and put it into our memory bank as well. So this is also called the humoral immune response. Again, it's going to be when the B cells or the bone cells are responding to foreign invasion. It's going to do so by creating the antibodies that are secreted, and those antibodies are going to then bind to the cells that are going to have the antigens, targeting them for destruction. Activated B cells are able to bind to any of the antigens that they have had, basically they've been targeted, so any unprocessed antigen that they already have their target against or the antibodies against. And once antigen particles enter the B cell by endocytosis, then we can create antibodies against them. So once we endocytose this unprocessed antigen, then we process it using those macrohistocompatibility complex proteins, then we are going to then use them as an antigen presenting cell. So remember, the B cells then also antigen present. So they take what they've ingested, they break it down into small components, and they present it on the outside of their cell, again giving the bloodhounds the sense of whatever the foreign invader is that they're trying to attack. Helper T cells are able to recognize a specific antigen that's bound to that complex and then causes the release of interleukin-2 or IL-2, which, as we know previously, is going to increase the immune response as well. So this is what B cell activation looks like. So here's a B cell. It's going to find a foreign invader like a pathogen. So these little red cells are the antigen on the surface. It's going to phagocytose that pathogen that has antigens in it. And then once that pathogen is degraded or digested, we're going to take the antigens and present them on the surface, again with that macrohistocompatibility too. And what happens is we're going to get a T cell response. Okay, so a T cell dependent response is going to be a strong response, and a T cell independent response is going to be a weak response, but both of these are going to create an increase in the amount of antibodies that are circulating through our bloodstream. So the strong response is going to be when the T cells are going to have the 8s, right, are going to have a nice strong reaction with that antigen, creating more and more of these memory cells as well as plasma cells that create antibodies. The T cell independent response, although it's a little bit weaker, is going to be faster. It goes straight to the end game here, which is creating antibodies from the plasma cells. Okay, anyway, long story short, the end goal here is the secretion of antibodies, which then are released into the bloodstream to find this pathogen, or the brothers of this pathogen. This one's already been degraded, but we're going to find the brothers of this pathogen and attack it by targeting it with antibodies, which release basically red flags for other immune cells to come and degrade that pathogen. Okay, so this is what an antibody looks like. An antibody is going to have these large and small IgG fragments. These have variable regions, which are going to be different for whatever it's binding. And then these constant regions, which are the same for all IgGs, IgA, whatever type of Ig it is. All right, and by binding, or by having this variable region that has a specific binding response to this antigen, now remember this antigen is on the surface of a pathogen or some sort of foreign invader, so now when these antibodies go out in circulation, they can bind to the surface of the cell that's presenting the antigen and target it for destruction. Okay, so here's what I mean by that. So here we have the antibodies. The antibodies are going to go out into circulation. They're going to cause an agglutinization reaction where they're binding to the antigen on the surface of the pathogen, which sends up a red flag for something like a macrophage, which is able to then phagocytose this foreign pathogen. Same thing with the natural killer cells. Instead of phagocytosis, however, we're going to bind to it with the antibodies that are actually on the surface of the natural killer cells, as opposed to like circulating through in suspension in our blood supply. Natural killer cells have that antibodies right on the surface. Same thing, though, we're going to bind to the antigen, again on the surface of the pathogen, and then release perforin. Perforin is going to cause lysis, and that's going to then obviously cause the cell to die. So cytotoxicity, or cell death. Okay, so again, as I mentioned, we have memory cells, which serve as the ability to basically track your arsenal of everything that you've ever utilized to attack any foreign invader over your entire lifetime. Okay, so the first time that you're infected, You're going to create a clone of the lymphocytes that are able to recognize that pathogen. Again, T cells and T cells and B cells make memory cells. So the next time that you encounter that pathogen, you have a far more effective immune response. You already have the weapon that's very specifically targeting that particular pathogen, and that secondary immune response is so strong that you typically don't even know that you fought that pathogen. That pathogen comes to the door, you go boom, see you later, and you don't even give the pathogen enough time to reproduce underneath the surface of your skin, right? So the secondary immune response is extremely strong and allows us to have a resistance to the second time or the subsequent times that we are exposed to a particular virus or sickness of some sort, right? Virus or microbe, etc. Okay, so these are different types of adaptive immunity. Again, adaptive immunity is immunity that you have acquired as opposed to innate immunity, which is basically just a broad sweeping ability to kill any foreign invaders. So adaptive immunity can be either naturally acquired or artificially acquired, as well as being either active immunity or passive immunity. Okay, so this is what we were talking about previously, naturally acquired active immunity. This is basically when we have been previously affected and we create memory cells so that the next time we see that infection, we just kill off that foreign invader. Now, we can also naturally acquire antibodies through what's called a passive response. Picture, for example, that a mother who is breastfeeding has been exposed to, I'm just going to make up a number. Let's say she's been exposed to 100 different infections throughout her lifetime. It's probably a really low number. It's probably closer to 1,000, right? And she has created antibodies against all 100 of those infections. When she creates her breast milk, she is going to secrete those antibodies into her breast milk during breastfeeding, which can cause an infection response or a halting of a temporary halting of an infection response inside the baby. So any infection that the mother already made antibodies against, the baby might not fall sick to during the breastfeeding time frame. So that's called naturally acquired. Again, it's acquired naturally because you are going to take it in from breast milk. And it's a passive immunity in that you didn't have to make the antibodies yourself. You were given the memory cells and they were passed on to you. Again, this is going to be a temporary prevention of infection because typically these memory cells don't last for a very long time, but it does allow the baby to become immune essentially to anything that the mother has seen throughout the lifetime. Okay, so let's talk about artificially acquired immunity. We have artificially acquired active immunity, which is when we are going to, for example, a vaccination. We've had a shot in our arm of an infectious organism, which has usually been weakened or is just a piece of that infectious, so an antigen of the infectious organism, but we are still going to have that antigen, and so we're going through the antigen presenting response. That stimulates the immune system to produce memory cells, which are going to then be able to attack the disease-causing cells if you were to see them, but you haven't seen them yet. You've only seen what they're going to look like. So this is the equivalent of the bloodhound being given the scent before you've ever been exposed to the disease. Okay, so again, that's vaccination. Now we also have artificially acquired passive immunity. Picture that you get a blood transfusion from someone who is going to have antibodies from whatever it is that they have ever been exposed to. That's called artificially acquired passive immunity. So just like getting the breast milk from someone, that's a natural acquisition, you've artificially acquired it by transfusion, but it's going to provide a temporary immunity specifically related to the injected antibodies. So people who have issues with immune responses or have people who have basically any sort of immune disease can oftentimes get relief by getting blood transfusions or plasma transfusions from individuals who have normal immune responses because they're able to then temporarily get anyway, some of the antibodies from that individual, which helps them fight off or stave off any of the foreign invaders that might attack them. All right, so let's talk about vaccines for a little bit. And I want to be really clear here that when I'm talking about these vaccines, this is not the COVID vaccine. The COVID vaccine is completely novel, and it is going to be created using mRNA, which is a completely new way of vaccination. So that is not what I'm talking about when I'm talking about how these vaccines are made. But overall, vaccination happens when we introduce something into our body that is going to be an inactivated version of whatever our foreign material is that's going to stimulate our immune response artificially without actually seeing the foreign invader. We'll see a piece of it, which allows us to increase our immune response. So the vaccination is going to hopefully trigger the immune response without causing an infection itself. Okay. There are different types of vaccinations. As I mentioned, the COVID vaccination is an mRNA vaccine. We're not going to talk much about that today. But we also have live viral vaccines. So live viruses are going to be prepared when we have a weakened or attenuated strain. So it's going to be devoid of pathogenicity, but is still alive. So it's been really, really weakened and beaten down, but it's not just a piece of the virus, it really is the whole virus. And that gives us a protective immune response. So basically what we can do, some of the first examples of this, we're using something very similar to smallpox, such as cowpox, but it's a much more benign virus. It doesn't get you as sick. And once you have a immunity to cowpox, you have an increased immunity against smallpox as well, because they're very similar. Now, one of the more common examples these days, for example, almost everybody here has probably had this vaccination, is the childhood MMR vaccination, which prevents against measles, mumps, and rubella. And this is going to be a way that we are going to inject pieces, again, of the virus, or we're going to inject live viruses that have been weakened into an organism, into an individual, whose then immune response is able to mount against the viral particles and creates an immune response when they do actually see the live virus. Most of the vaccines that we get, however, are what we call killed vaccines or particle vaccines. So when we have destroyed most of the proteins that are going to cause the virus to function, but we're going to leave the proteins on the outer shell intact because, again, this is what our immune system is going to see. So non-replicating virus vaccines are going to have a lot more antigen than live vaccines. Why? Because live vaccines actually create the immune response, whereas non-replicating viral vaccines are going to kind of induce a pseudo-immune response, if you will. And we're basically going to be injecting the individual with a non-infectious version of the virus and causing a reaction within the immune system, hopefully, to jumpstart the immune system so that when we see it again, we already have the memory cells to attack it. Some other types of viruses are toxoids. So these are toxic compounds that are inactivated, but they're similar to the microorganism itself. So instead of giving you the actual microorganism, we give you toxic compounds that the microorganism produces that cause illness, so that when we start determining that we have these microorganisms or the compounds that the microorganism produces, we're able to actually realize that we are infected and go and attack it. Some examples of these are going to be our tetanus, pertussis, and diphtheria vaccine, the DPT. And this one is going to be the kind of vaccine that we're going to need to take multiple times throughout our lifetime. Multiple doses usually on the first vaccination attempt, so 6 months to a year apart or whatever, and then also a booster shot in our teens. Sometimes we utilize recombinant peptides that are going to be pieces of viral proteins. These pieces are not actually going to be able to put together into a virus itself, so there's zero worry of infection from the entire pathogen because, again, we just have pieces of the pathogen. And by exposing us to the pieces of the pathogen, some of the immunogenic proteins of the viruses, like glycoproteins, etc., are not going to cause as much of an immune response as before, but they are still going to cause the immune response. So good examples of this are going to be the recombinant hepatitis vaccine. We're using an adenovirus vector, so we're not using hepatitis as the vector, but it looks like the hepatitis. And another example here is the AstraZeneca COVID-19 vaccine. Many of you may remember that AstraZeneca went with the old school style of vaccination, and Johnson & Johnson blew it out of the water, and Johnson & Johnson right now is one of the lowest effective rates, right? So Moderna and Pfizer, all of those have much better efficacy. So although we did end up with the AstraZeneca vaccine, it didn't seem to be one that became very popular. But it does work in the more traditional mechanism by exposing us to specific peptides of COVID-19 instead of the mRNA, which is what we utilize for the other vaccinations. Now, adenovirus itself is going to cause an immune stimulation. And then once we have the immune stimulation, again, we're using adenovirus as a vector, we're then going to, because the immune system is overstimulated, then it's going to attack the pieces of the recombinant hepatitis C so that we can create an immune response, again, against something that have not yet been exposed to, so that if we are exposed to it in the future, we can kill it very quickly. All of this is important, and the importance of all this is to make sure that our dosages are correct, because we don't want to get too sick, right? Not too much adenovirus, but we want to have enough adenovirus that we can stimulate an immune response. Okay, so again, this is kind of like how this works. So here's a recombinant DNA vaccine. So we're going to extract the gene from Hep C and pop it into a different gene, so into an adenoviral vector. So we're going to take the red part out with restriction enzymes or molecular scissors, snip that out, and pop it into an adenoviral vector using, again, restriction enzyme digestion. And then once it's in there, we pop it into the adenovirus, where it replicates very quickly. So the adenovirus now has not only adenovirus, but also a little piece of hepatitis C, so that when we get sick from adenovirus, this is why after you get a shot, you might feel sick for a couple of days. When we get sick from the adenoviral vector, it increases our immune response. And while we have an increased immune response, then we make antibodies not just against the adenoviral vector, but also against hepatitis C virus. So that's how recombinant DNA vaccines work. Now, some of the more modern vaccines, the COVID vaccines, are going to be mRNA created. And these are very, very specific, which means they are highly effective. But because they are so specific, it means that they are not necessarily effective against multiple strains of the virus. So it's an mRNA sequence that we've created basically to produce the spike protein found on the surface of our own cells. So we have a COVID-19 spike protein, and then we're going to use that mRNA. We isolate it and engulf it in lipid nanoparticles and inject it so that our cells are able to pick up the mRNA nanoparticles. And then we're able to stimulate the immune response by attacking, basically, not attacking, but recognizing what that mRNA looks like and utilizing that as what we're going to rise an immune response against. Now, because these are so incredibly new, in fact, this is the first time we've ever used mRNA vaccination rates, we don't know the long-term effectiveness. That's why they keep going back, oh, you need 2 shots, now you need a booster, now you need 2 boosters. If you're elderly, you can get a booster every year, because we don't know how often we have to continue to give this mRNA. We don't know really how long it's going to last, but we do know that it's highly effective in preventing sickness, or at least preventing extreme severity of symptoms if we do get sick. All right, so again, here's the virus, the COVID-19 RNA. It's an RNA virus. Coronavirus is going to be an RNA virus. And we have these spike proteins that are found on the outside. So we're going to take that spike protein that's found, again, on the COVID-19 virus and inject it, and the mRNA for that protein anyway, into a lipid nanoparticle. So when we're injected with that lipid nanoparticles, those are going to end up engulfed into our cells, and then we can create antibodies against the COVID mRNA. Now, again, because it's so novel, we don't know how long it lasts, but we do know that the efficacy is incredible. All right, so what happens when the barrier of self versus non-self breaks down? So this is when we get what's called an autoimmune disorder. Again, this is when the immune system is going to attack its own self. And this is because of something called self-tolerance. So when self-tolerance goes awry, we don't know what is self and non-self. So in order to correctly differentiate between self and non-self, we go through a negative selection process. Again, that's for self-tolerance, where any receptors that attack self proteins are eliminated. So if you attack your own cells, we're going to destroy you. Then we have another region where we're going to basically, if you have an autoimmune problem, you can have B cells and T cells that bind to self protein as though it were a foreign antigen. So something that should have been eliminated is not. And when it is not, then the body, the immune system, attacks self. It's not sure exactly why this happens. There could be environmental factors involved, etc. But we do know that these individuals are going to have a series of treatments to help create more self-tolerance. Now, autoimmune diseases typically happen after puberty, and it often happens to women. Over 2/3 are going to be women. We don't know exactly why autoimmune diseases are caused. We don't really have cures, but we do have treatments, and the treatments are going to involve immunosuppressing medication. That means that individuals that have autoimmune disorders are more likely to get sick because they are immunosuppressed by design to keep their body from fighting itself. That means that these individuals need to be really careful of exposure to things like COVID-19, etc. So this is a subpopulation that might not be able to get that vaccine because the vaccine might not work as well in individuals who have autoimmune disorders, or it might cause an attack of self. So we intentionally try to avoid immunization of individuals who have autoimmune diseases, which means that they have to self-isolate from the rest of the population during this recent pandemic. Okay, so autoimmune diseases. We have over a third of the population diagnosed with some sort of autoimmune disease, and we know that autoimmune disease prevalence has been increasing on an exponential rate over the last 50 years, particularly because a lot of these are diseases that were previously not treatable, and now they have become treatable. Things like celiac disease, which is dietary restrictions, diabetes, which is now treatable with insulin, etc., whereas it wasn't previously treatable, so these individuals might die off before they pass it off to their offspring. But at this case, at this point, because we're able to treat them, these are going to be on the increase or on the rise. So autoimmune diseases have been increasing over the past couple of decades. These are some examples of common autoimmune diseases like multiple sclerosis, diabetes, celiac, etc., and the different tissues that they can affect. Rheumatoid arthritis is a pretty common one, affecting the joints. For example, ulcerative colitis and Crohn's disease attacking the gastrointestinal tract and intestines. And autoimmune diseases have had a lot of research, but they don't really have any. There are not a lot of things that we know for certain. So we do know that there's a correlation between the rate of infections and autoimmune diseases. So in areas where infection rates are pretty high, autoimmune disease is very low. So attacking self versus non-self doesn't seem to happen as much when there's actually something for your body to fight. So when you are actively attacking non-self, you are less likely to actively attack yourself, if that makes sense. And also, this has been correlated to a exposure of bacteria concentration. So if we have less exposure to beneficial bacteria during childhood, then we're going to have less normal flora inside our gut. Same with children who have had antibiotics prescribed during childhood, it's going to kill off normal flora. Then sometimes, because we don't have that normal flora, we're going to have an increased risk of asthma and allergies and other types of autoimmune disorders. Okay, sometimes the immune system is going to cross-react. So sometimes the immune system is going to cross-react with self-tissue after seeing an infection. So just like we said that cowpox can cause memory cells that are also going to cause protection against smallpox, sometimes a tissue that is non-self will look very similar to self. And so once the antigen is presented, then the B cells go around and might attack self-tissue after fighting a certain infection as well. And as we mentioned, gender tends to play a large role in this. Women tend to have have an increase in the autoimmune disorders. We're not exactly sure why that is. Perhaps differences in hormonal balances or in the immune system, but we're not able to isolate any particular factor here. But we do know that men are not as highly affected as women. There's also genetic factors, as well as the concentration of particular biomolecules like vitamin D. Remember, vitamin D, we make a precursor. It's going to lay underneath the skin, and then UV light is going to cause it to become converted into its final form. So we do need UV light exposure. So if we have more UV light exposure, that can increase our immune response and give us less autoimmune disorders. So we do know that some individuals who have autoimmune disorders, if they have exposed to UV lamps or whatnot and get an increase in vitamin D, or like supplementation from vitamin D, for example, it can decrease but not reverse the autoimmune disease. So again, it's probably multiple factors involved here. And I would be remiss if I talked about the immune system without touching on allergies. We talked about the histamine response and how it's going to bring over different types of cells from the immune system to an area that's been infected. And allergies are basically when we cause a reaction to a harmless antigen as though it were a pathogen. So it's also going to be antibody-mediated responses. They're called hypersensitivity reactions. And what do? We give them an antihistamine to help slow this reaction down, right? So asthma is a particular type of allergic reaction, which is going to cause an excessive histamine release. Histamine release is going to cause constriction of the airways, which causes an inability or decrease in inability of this individual to be able to catch their breath or to breathe properly. Okay, allergic reactions are going to be responsive to the immunoglobulin IgE, which is going to bind to other immune cells like basophils and mast cells, which are then going to, again, release histamine and other types of cytokines. This is going to be an inflammatory response, which causes vasodilation, bronchoconstriction. Again, this is going to be something you see in asthma, right? Constriction of your bronchioles, as well as smooth muscle contraction, the creation of mucus, and watery eyes or sneezing or itching. All of these are your typical allergic reactions. Now, a systemic allergic reaction is called anaphylaxis. An anaphylactic shock can cause the entire body to go into a shutdown, essentially. And this can happen depending on what you're allergic to, right? How your immune system responds to that type of allergen and how that allergen has been presented to the body. So someone who's severely allergic to bee stings, for example, without that EpiPen was going to go into anaphylactic shock, be unable to breathe because their throat is going to close, and eventually end up suffocating, right? So we want to make sure that anybody that has a severe allergic response or knows they have a severe allergic response potential is going to carry an EpiPen on them at all times. Now we have specialty allergies like food allergies and allergies to venoms, like I mentioned, bee stings, but severe food allergies as well. These guys can be much more severe than inhaled antigens and can be lethal. So what happens after we're exposed to an antigen? Okay, so here's the allergen that enters and the antigen that enters into the body. It's going to bind to the B cells, the antibodies on the surface of the B cells, and then that's going to process that antigen and now create antibodies. So once the B cells create antibodies against that, in this case an IgE molecule, they're going to secrete those antibodies and that are going to help cause an immune response against that allergen. The IgE antibodies attached to mast cells here, and the mast cells have these histidine granules, which then are going to get released. And once we release these, that's going to cause other substances, etc. So the histamine release is what causes what we just talked about, that allergic reaction itself. All right, as we get older, what happens? So some of the effects of aging include a decrease of your lymphoid cell production. So you're going to create less B cells and less T cells. Again, these are involved in the immune response, so a lack of them can cause an increased infection rates. Also, again, if we have less cells and we have less ability for the cells to proliferate, so we're going to reduce them. Additionally, our thymus, which is where our T cells come from, becomes less and less proliferative and becomes more and more fat tissue. So over time, it's going to become less and less productive. We also are going to increase the apoptosis of our immune cells, so they start to die off. We're going to increase the production of regulatory cells as opposed to Th1 and Th2 cells. So these are thymal cells that are going to, again, be responsive for the immune response. And eventually we can enter into chronic inflammation. So if we end up with areas that have just constantly been under inflammatory or stress, can cause exposure to toxins and DNA mutations. And our endocrine functions are also going to affect our immune system. So as we get older, our endocrine system can start to decline. And so if we're producing less growth hormone, et cetera, that can also cause our immune system to start failing. All right, so this here is the answer to the application question. Harold has been invaded by the common cold. And so the activation of the innate and acquired immune systems have begun. In the innate response, we have non-specific immune cells like NK cells that kill the viral infected cells, macrophages and dendritic cells that are also phagocytic and are antigen presenting. And then they're also going to stimulate interferon in the innate response. We also have neutrophils present. These guys are going to be in the area of inflammation. They're going to be coming out or extravate out of the blood supply and then helpfully kill these different viruses. So the immune cells go out and kill the viruses. But we also have the acquired immune response, where we have that APC or antigen-presenting cells that are going to go to activate helper T cells. APC secrete IL-1 and helper T cells secrete IL-2, and then that's going to cause the proliferation of T cells. We also are going to have helper T cells differentiate into Th1 cells, which are going to be cells that actually attack. So they prompt the cytotoxic T cells to attack the virus-infected cells. And last but not least, we're going to be making memory cells of both our T cells and our B cells so that the next time that we see that exact same cold virus, we're not going to fall sick to it again. So we're going to have a secondary immune response, very fast and rapid response, hopefully killing the infection before the microorganism can reproduce and take hold. All right, thank you so very much for attending class today. I really appreciate your time. Good luck and happy studying. Have a great day.